Hi, my name is Fleicy Wang, and I am a rising senior at Palo Alto High School in Palo Alto, California.
Hi, my name is Iona and I'm a rising sophomore at Monte Vista High School in Cupertino, California.
Hi, my name is Nee, and I'm a junior at Yerba Buena High School in San Jose.
Hi, my name is Priya Iyer, and I'm a junior at Evergreen Valley High School in San Jose, California, and we're working on SIP project ENV-01.
Our research project is called Do Evolutionary Relationships and Environmental Conditions Influence Plant Traits in Coastal California Ecosystems?
Here is our presentation outline. We're first going to talk about some background information, including restoration and climate change and traits in phylogenetics. Then we're going to move on to our research questions and hypothesis, study sites, and sampling methods. And finally, we're going to talk about our results, discussion, and future directions.
Restoration is a growing practice where ecologists seek to restore damaged, destroyed, or degraded ecosystems. For instance, the image on the right shows an example of the restoration process, where from 2008 to 2011, the ecosystem was recovered to its former state. In restoration ecology, there are several factors that must be considered when seeking the ideal recovery strategy. For instance, the natural recovery rate or ecosystem resilience, past land use, and the surrounding landscape context must all be taken into consideration. In addition, it's also necessary to ensure that there is enough designated funding for the restoration strategy being employed, and that such a restoration strategy aligns closely with the desired goals of the project. Since our project hopes to ultimately improve or restore the conditions of California coastal ecosystems, it's really important to have a basic knowledge of the restoration processes, and we hope that by analyzing the functional traits of certain plant species, we can improve the chances of such restoration success.
As humans continue to damage the earth by the use of fossil fuels, the emission of greenhouse gases continue to increase. This causes the earth to continue to get warmer in a process called global warming. As the earth gets warmer, weather becomes unpredictable. There are more droughts and a variety of other weather issues causing climate change. This then leads some plants to not have enough water to grow due to droughts. This is part of the environmental variability that affects plants.
So what are functional traits? Functional traits are characterized as both response and effect traits and are simply defined as traits that either addresses the responses of species to changes in the environment or the species contribution to the function of the ecosystem. They are extremely important as they relate to the responses of a plant's survival and also their reproductive output. And by measuring and understanding these traits, it helps us to understand restoration better as it increases our knowledge of ecological process.
Response and effect traits are in a big category of functional traits. The difference between response and effect traits are all based on the environment. Response traits respond due to the environment. For example, the growth rate of the plant is based on whether there is enough resources in the environment. Effect traits affect the environment. An example of this would be the way a tree's shade will affect its surroundings. This will then affect its ecosystem functioning or its resilience in the ecosystem. We are specifically focusing on response traits in our research.
A phylogenetic tree is a diagram that represents evolutionary relationships among organisms. They are often measured in millions of years ago. The pattern of branching in a phylogenetic tree reflects how species or other groups evolved from a series of common ancestors. For example, if you look at the joint in the tree, it shows the last common ancestor that these two species evolved from.
The research question that I'm hoping to answer in this project is, how do evolutionary relationships affect leaf lobedness between plant families? My hypothesis is that plant families will be more predictive than genus to determine differences in species traits.
My research question is, are climate variables correlated with the Pacific leaf area? And my hypothesis is Pacific leaf area will be greater if the vapor pressure deficit is greater.
My research question is, how similar are the specific leaf areas of the same genus compared to the specific leaf areas of different families? And my hypothesis is that the specific leaf areas of the different genre will be more similar than that of different families.
My research question is, how does climate affect specific flower area? My hypothesis is that specific flower area will be related to mean annual precipitation.
On the right is a map of all the study sites that we gathered leaf samples from. They include all the coastal counties from Humboldt to Los Angeles, excluding Ventura County. The entire range is approximately 1,200 kilometers, and we sampled between coastal grasslands and prairies, coastal sage scrub, and coastal dunes. There were a total number of 34 study sites, of which we obtained from a total of 10 different counties. The total number of plant species that we collected was 40, including species from a total of 9 different plant families. Of those 9, we primarily focused on the Fabaceae, Asteraceae, and Poaceae plant families. Here are some images of the plant families that we have been working with: Acnospon strigosus, Agrostis pallens, and Grindelia camporum.
Due to remote internship, we have been using a software called ImageShade to measure plant functional traits. The functional traits that we have been studying this past summer are listed above in the slide. In this presentation, we focus on the bolded traits, so Pacific leaf area, Pacific flower area, and leaf lobeless. Pacific leaf and flower area are the fresh area divided up by the oven dry mass. Leaf lobeless is the ratio of perimeter to area multiplied by the ferrite diameter. Pacific leaf area is important because it relates to photosynthesis and the leaf lifespan. Pacific flower area is important because it relates to pollination and seed production. And finally, leaf lobeless is important because it relates to the leaf energy balance and how it can help regulate leaf temperature.
We track weather data for each of the sites that the leaf samples are from. Each location has the latitude and longitude listed, as well as the coordinates of the weather station that corresponds with each site. The distance to collection is the distance from the weather station to the site that the leaf sample is from. All of the other variables, precipitation, temperature, and humidity, are data for a particular station for the month and/or year that the leaf sample was collected. We only collected climate data for the months of April, May, and June, because those are the months that are in the peak growing season for coastal ecosystems in California. In order to test our research questions, we tested functional traits against other variables. If a p-value is below 0.05, that means that the traits correlate. Here, we tested whether specific flower area was correlated to different climatic variables: annual temperature, annual precipitation, annual vapor pressure deficit, and vapor pressure deficit during various months in the growing season. The y-axis is the log transformed value of the specific flower area, and the x-axis is the corresponding climatic variable that is being tested. We found that annual temperature, annual precipitation, annual vapor pressure deficit, and vapor pressure deficit from all months except for May were not correlated to specific flower area. However, the vapor pressure deficit in the month of May was weakly correlated to specific flower area.
Here we tested whether Pacific leaf area were explained by the different climate variables, such as the mean annual temperature, the mean annual precipitation, and the vapor pressure deficit. For each of these graphs, we have the log of SLA on the y-axis and the corresponding climate variables on the x-axis. As you can see, the first 3 climate variables, mean annual temperature, mean annual precipitation, and the annual vapor pressure deficit, does not have a correlation with the SLA because their p-value is above 0.05. However, we did find the average April VPD to be weakly correlated with the SLA. The higher the VPD was, the higher the SLA tended to be.
We also tested to see if evolutionary relationships played a part in specific leaf area, which is a commonly used functional trait for plants. The top 2 graphs show the absolute value of the specific leaf area, and the bottom 2 graphs show the trait variability, or basically the coefficient of variation of the specific leaf area. The left shows the differences in plant family, while the right shows the plant genre. For the absolute values, there appears to be little difference between the plants, with there being no difference between the different plant genre. For the plant families, there's also little difference, with there only being differences between Poaceae and the other plant families. Poaceae had significantly lower specific leaf area than the others. For the trait variability, there's no difference between both the plant genre and plant families.
Here we tested leaf lobeless with the different climate variables. For the mean annual temperature, and the June average VPD, we find that they both have a weak and negative correlation with the leaf loadness. And for the annual precipitation and the annual average VPD, we find that they both have no correlation with the leaf loadness.
We also tested to see if evolutionary relationships played any role in the values of leaf lobedness for each plant family and genus, with plant family on the left and plant genus on the right. Some of the absolute trait values were found to be significantly different between plant families. For example, Poaceae is primarily different from Asteraceae and Fabaceae, while Fabaceae is also different from Papaveraceae. When looking at the absolute trait values by genus, there are also significant differences, such as between Acmaspon stipa and Festuca. When comparing the two graphs, it is also evident that grasses, which are in the yellow, tended to have higher leaf lobedness, with Festuca and stipa driving the larger leaf lobedness values. We also tested for the coefficient of variation, which estimates trait variability, but no significant difference was found between plant families. However, one significant difference between genera was found between Acmaspon and Baccharis.
Specific leaf area is often used to relate plant distributions to climate, but we found that specific leaf area was not related to most of the climate variables that we tested for. We also found very few differences between specific leaf area and taxonomic classification, like family or genus. We found specific flower area was related only to vapor pressure deficit during the peak bloom season. The relationship was positive, which indicates that specific flower area will increase with higher temperatures. This could indicate that plants are attempting to compensate potential phenological shifts from higher temperatures with investing more energy into floral development to potentially attract more pollinators in a shortened flowering period.
Currently, most ecological studies that use easy to measure or quote-unquote soft traits focus on specific leaf area. However, we found that specific leaf area of herbaceous plants in coastal California were not consistently related to climate or taxonomic classification, unlike leaf lobedness, which could indicate that functional-based plant restoration or conservation should further consider less commonly measured soft traits. This is especially relevant to coastal California ecosystem management because other studies have found that leaf lobedness was important in predicting the mortality risk of restoration plantings.
There are still some questions we have for the future. Firstly, do the patterns we found hold true in non-coastal habitats, or will we incorporate additional plant families? Our study focuses on coastal environments in 3 major plant families, Asteraceae, Fabaceae, and Poaceae. But California is a very diverse geographic region, and there are non-coastal habitats and many more plant families to study. Also, how are reproductive traits related to leaf traits? There are many studies on leaf traits, but not on reproductive traits. Linking reproductive and leaf traits could be useful in ultimately finding out more about plant success.
We would now like to acknowledge our mentors Justin Leung, Dr. Michael Lloyd, Dr. Kathleen Kay, and Dr. Greg Gilbert for their inputs, in addition to the SIP program and our families for their support. We would now like to open up for any questions. Thank you.
Thank you, guys.